welcome to the first of this year's Dean's Research Seminars in the Faculty of Veterinary and Agricultural Sciences. I guess just about afternoon, and there'll probably be a few more people joining, but I think we'll get started. So before we begin with the seminar proper, I'd like to acknowledge the traditional owners of the land I'm on today, which is the Boomerang people, as well as the traditional owners of the land you're situated on, and acknowledge that Indigenous Australians have been the custodians of the lands and waterways of Australia for thousands of years, and I pay my respects to their elders, past and present. Today, Professor Kevin Smith from the School of Agriculture and Food will present the Dean's seminar, which is Converting Genomic Discoveries into Genetic Solutions, Better Pastures for the Australian Dairy Industry. A little bit of background on Kevin. Professor Smith earned his Master in Agricultural Science and his PhD at Melbourne University, and is now a world leader in pasture plant genetics and breeding. He's worked in both government and private sector agencies and has been actively involved in developing biotechnology and innovation projects and strategies, and collaborating with academic and industry partners across Australia, New Zealand, the UK, the US, China, Argentina, and Uruguay. He was the Deputy Director of Bioscience Research and the statewide leader for plant breeding at Agriculture Victoria Research before joining the university in 2012 as a professor in pasture agronomy and pasture breeding. He still, though, works with Agriculture Victoria Research at their premises, and it's from there that he will join us today. With over 100 peer-reviewed papers and over 500 conference papers to his credit, Kevin's research interests are in plant breeding and genetics, with a particular interest, as I've mentioned, on pasture grasses and legumes. His work predominantly takes place in partnership between Agriculture Victoria and Melbourne University, and he's supported by Dairy Australia and the Gardner Foundation. He's supervised or co-supervised 23 PhD students to completion and currently supervises 5 graduate researchers. Please welcome Professor Kevin Smith. Kevin, over to you.
Thanks, John, and thanks for that introduction. So, as John said, I'm going to share a presentation with you today that covers some of our research activities, and I hope you can see the PowerPoint now.
Yep, we can see that fine, Kevin.
Thank you. So what I'm talking about is the work that we've been doing really that is about taking the era of genomic breeding into pastures and ensuring that technology makes its way to industry. Ultimately, the farmers, and in many cases dairy farmers, are our primary target, but also we work closely with seed companies to ensure that the discovery and technologies that we do actually migrates to that next end user. And what I'll do with a couple of case studies is really demonstrate how that partnering and joint work has influenced the kind of science we do and how we go about ensuring that we have impact. So, just waiting for a slide to advance. There we go. So, unlike some areas of science that we work in, forage breeding is actually a relatively young science. And the first scientific breeding of grasses was really pioneered in the Welsh Plant Breeding Station in Aberystwyth in the 1920s and 30s, and also in other locations in Europe and the United States. And from that beginning of time, the emphasis really was to look at ways to improve the productivity and sustainability of grasslands so that they could support improved animal production. I'd also like to point out that our own faculty had a pioneering researcher in this area, Dr. Yvonne Aitken, and I was very fortunate to commence my undergraduate career at a time when Dr. Aitken was still active within the faculty, and her work that was really around pioneering the impact of climate and environment on the expression of flowering time in a range of crops, but particularly ryegrass initially, is something that I still use in my research every single day today, as we use that to work with new F1 hybrid technologies where we're crossing perennial ryegrass And so we have that basic underpinning science that has led to us being able to use genome sequencing to develop a hybrid system in a species which doesn't regularly hybridize. So I think, again, sometimes we don't necessarily see when the impact of our work will be seen or the enduring legacy of that. A typical forage plant breeding program has taken around 10 to 30 years. So from the time that a breeder such as myself and my early career was as a breeder is defining the traits and making the initial crosses, they then go through a period of seed production of multiple generations so that the traits become stable within that population. And most forage species that we use in Australia are actually outcrossing, and many of the aspects of science that we use in breeding are more similar to animal breeding. And you can think that the bag of ryegrass seed you buy or the bag of lucerne seed you buy with a variety name or a cultivar name actually contains as much genetic variation within that variety as we might find in a breed of animals. So there's as much variation in a usual lucerne cultivar or ryegrass cultivar as there is in the Aberdeen Angus breed of beef cattle or Holstein Friesian dairy cattle. So there's plenty there for breeders to utilize. And ultimately then, once we've done this initial evaluation, there's a lot of client-focused evaluation where we go under grazing and we look at how animals interact with the plants that we're breeding before they're finally evaluated. So what you can see there is that it takes a long time to go from an idea to an outcome. And so what a lot of our work has really been focused on ensuring that we get better varieties and that we get those varieties faster, to look at trying to change that generation interval. Seed companies, of course, may wish for a variety to stay in the market for a long time to maximize their return, but we really want to see the best genetics getting onto farm in an appropriate way and as quickly as possible. So whilst I'm passionate about plant breeding, there are some inconvenient truths that I think we need to face, that plant breeding is slow. Plant breeding in the modern context is big. And by big, I mean that the scale of the work to achieve the genetic gain is large. The number of collaborators that you need to take an innovation to the market is also large. And that's great from a science perspective. We get to work with plant physiologists, we get to work with the molecular geneticists, we work with plant physiologists. But we also need, as I've said, to work with the commercial sector and farmers themselves to really work and ensure that the products that we develop are appropriate for market. And this also includes the choice of technologies. So technologies such as genetic modification, which have been used and part of our science work, are not always acceptable in every market or every sphere. So we need to acknowledge that and think about that when we design our work. The other interesting aspect that is the selection environment, so the environment in which you do the breeding, will influence the way the trait's expressed and influence the rate of progress you can make. This is in many ways genetics 101 that people will have heard in their first or second year of undergraduate degrees in agriculture. But the significance of this in the modern world is even greater, because what we see is that breeding has become a multinational activity. And so large multinational companies are seeking to put much of their science into locations that suit their global marketing strategy. And it may be that they're not specifically targeting Australia and Australian farmers' needs when they make those selections. So again, by partnering with those companies, we can ensure that selection and evaluation is done in Australia, or we've done the science to understand that selections that are made in New Zealand or the United States or the Netherlands have a role here. The other thing that I've actually popped there is a statement that one of my genetics lecturers said many years ago, was you have to remember you get what you select for, even if you didn't mean to. And what we need to understand from that comment, and why I keep reminding myself of it, is that we often are selecting for an outcome, but we don't always know why we're getting there. So if we're selecting for yield, are we selecting at the expense of forage quality? So we have a very high yielding plant, but we've sacrificed animal nutrition. And I think this also is one of the drivers of the research of the teams that I've been involved with over the years, is to be more precise and develop accurate and quick tools to be able to measure more traits than the final outcome trait. And as I've already mentioned, market realities come to bear. So it is important that breeders understand this and have a strategy for getting that product ultimately out onto farms. I now want to talk a little about the science, and to set the context of modern plant breeding. And we're certainly in the era of omics. And we hear a lot of, and I really think there are 3 omics that we need to consider, and I'll discuss each of those today. The most recognizable of these omics siblings is genomics. And we've all heard about the use of genomics in a whole range of agricultural and medical spheres. So the understanding of the genomic sequence of an organism and the correlation of that sequence variation to traits of importance, be they disease resistance, be they productivity, be they associated with health and welfare outcomes, has really revolutionized the way that we see science and the way we can target interventions. So we can do that with a technology called genomic selection, which I'll touch on in this presentation as one of the case studies, where genomic selection is utilizing all of that sequence variation across the whole genome and massive computing power to take that variation, impute that variation, and be able to correlate that with field performance, to the effect that we can select an individual on its likely performance before we've actually done that. And this work was in agriculture largely pioneered through work in Australia and the United States, and one of our colleagues, Professor Michael Goddard, was fundamental in making the discovery in cattle work of how to design and implement this technology, and it has greatly increased the rate of genetic gain, and we're now seeing that happening in plant species. The final 2 concepts that I have on this slide, genome editing and genetic modification, are where we're actually making targeted interventions to alter the genome of an individual. Genetic modification has been around for some time, with the controversy that has come with that, but also the big gains in terms of the reduced chemical use and other things when we look at the Roundup Ready trait in corn and soybean, and the extent to which those crops now dominate that market. Be that good, be that bad, there's always 2 sides to every coin. But more recently, we're seeing genome editing, and genome editing is a more precise form of genome modification, where we're looking to be far more targeted, and sometimes we can do this in a way that we leave no non-host DNA behind. And that kind of site-directed nuclease work, where we're looking to target a gene and perhaps turn that gene off or remove that gene and leave no residue, is actually a really attractive proposition in forage plant breeding when we come to think about genes associated with lignification and forage quality, because we can knock out those genes, not leave any foreign DNA behind, and do that in a way that's more amenable to regulation. We then have phenomics, which will be the major part of my presentation today. And you may or may not have heard of phenomics, but it's really a modern version of plant phenotyping. And what we're looking at here is how we measure plant form and function using sensors, drones, and robotics. And I say it's the youngest of the omics siblings because it's really something that we're pioneering now in terms of, can we measure these traits faster, cheaper, and more often to be able to design better programs? And I guess the one that most people wouldn't expect the geneticist to be talking about is economics. But in the latter phase of the work that I'm doing now, it's become very clear to me, and we work actively with a team of economists, because if things don't stack up economically, then they just don't get to market. And so that economic stack up can be whether a seed company can make some money from it. So that looks at costs of regulation and the burdens of regulation. But most importantly, it's about what's the likely benefit to a farmer of adopting this technology and trying to find ways of valuing that genetic gain. So this graphic here really shows that what we're about is genetic gain. So we're about saying, how do we improve the baseline performance of a variety? And genetic gain is like compound interest. You can lock it in and it just keeps growing. So even if the rate of genetic gain is reasonably low, if you compound that, eventually you make large change. But as my title and introduction for this seminar suggested, the rate of genetic gain in forages has lagged behind that of major food crops and animals. So we've seen rates of genetic gain in the order of 0.5% per annum, whereas in the major food crops, we're often at 1 to 1.5% per annum, and likewise animals. So you can imagine that that creates challenges in terms of getting investment in this area of science, but it also creates an opportunity to use new technologies to bridge that gap and look at ensuring that, in essence, the pasture keeps pace with the rate of animal genetic gain, because if we see a gap in that, then animal industries rely on purchased feed to feed those animals to that high production potential. And often the cost of the purchase feed is higher than the cost of growing it yourself. So a whole range of factors come into increasing rates of genetic gain, but you can see that this graphic that I borrowed from Trends in Plant Science really looks at these same things in every crop and every commodity. We're around using high throughput phenotyping tools, so Genomics, better understanding the environments that we work in, and again, using these tools to achieve that. And I'll finally touch on how we can use genomic tools to breed for climates that don't even exist yet, such as in a context of climate change. So this slide here just shows that on one level, we have a whole range of traits which contribute to pasture growth, and you can quite think that we need to have plants that grow roots, have appropriate amounts of that, are tolerant to grazing, are efficient in the way they use water, flower at the right time to be synchronous with the natural rainfall, are able to regrow after flowering. So in one way, there's this really complex matrix of things that we could select for. But ultimately, you can describe at an output level the value of pastures against three main criteria: yield, so how much they grow and when they grow it, so which season it's in, the quality of that for grazing animals, and not surprisingly, for those that don't work in the area, the same attributes that are important for human nutrition or animal nutrition are expressed in plants, in pasture plants, as in some of our major food crops. So we see genetic variability for sugar content, for fat concentration. We see genetic variability for the amount of energy that can come from one unit of intake based on changes in digestibility. So by thinking of those outcome traits, we can start to make this complex puzzle more amenable to selection and improvement. So with all of that in mind, we started a program probably 15 years ago through the Cooperative Research Centre program to look at consolidating the science that underpinned forage breeding but particularly with perennial ryegrass as the emphasis, and with an emphasis on the dairy industry. So as the Dean mentioned in my introduction, this work has been part of a series of long-term programs funded by the Agriculture Victoria, Dairy Australia, Gardner Foundation and ourselves to really bring this science together and have that legacy of varieties getting out onto farms. The most recent iteration of that is a program called Dairy Bio, again with the same investors. And its, I guess, hero or highlight slide was around saying, we want to bring the technologies together to triple the rate of genetic gain that we are getting on in pasture plant breeding, to use hybrid technologies that I mentioned in the history slide around flowering time and manipulating flowering under controlled environments to get a 20% boost in yield. So if you think about that in the context of half of 1% per year, a 20% boost in yield compared to the base case is 40 years of breeding. So through this technology, we're given the equivalent of a breeder's whole lifetime of impact. To look at increasing the energy concentration of pastures by 2 megajoules, which seems like a low number, but you're looking at levels of 9 to 13 megajoules of energy per kilogram of pasture. So you're really looking at a significant increase in that, and to increase the persistence of pastures. So this is how long that they remain productive, remain economically viable before a farmer needs to go through the expensive and fixed cost of renewal. So I just have, as I mentioned, 3 case studies that I'll go through quickly for you, just to put some of the science we've been doing in context, and I'll start with economics. So one of the troubles with pastures and the economics of valuing genetic gain in pastures is we haven't had the tools to do that, and work that myself and a range of students and scientists from Agriculture Victoria, but also Bill Malcolm from the faculty have done over the years, is to really look at the paradigm of how we value genetic gain appropriately in pastures and contribute the component that is due to pasture to the productivity and profitability of the farm. And unfortunately, pastures aren't traded in a market, so they're an intermediary product, and we don't actually, they're used to produce milk and they're used to produce meat, but if we say that it's all of the value of pasture is expressed in that change of meat or milk production, we're actually overvaluing the value of pasture usually, which from my perspective as a breeder is probably useful, but it's not actually accurate. And the example I have here is, if you sell wheat, you know the value of wheat, you know how it's valued, and it's the same price for the same specifications, whether I'm an artisan sourdough maker and I take that kilo of wheat, turn it into flour, and sell my loaf of bread for $9 to $12, as if I'm a baker in an industrial bakery for a supermarket making a loaf of Wonder White that will sell for $1 a loaf. So same value of input, 2 very different products coming out of it. So what we've done is look at developing a system that says, what will these cultivar deliver in my system, and so what's the marginal value of the new genetics as opposed to the base case. And so we've done that in a range of work to value individual traits, such as increasing in energy or increasing persistence, but we've also been active in and leading a program to develop an industry-wide ranking tool for the dairy industry called the Forage Value Index, or FVI. So the FVI is the first time in Australia that farmers have been able to go online and get an objective rating of the likely value of forages in their environment that is based on both the yield and also the relative value of that yield in different seasons, so in spring, winter or summer. And this tool is hosted on the Dairy Australia website, and so anyone can go and look there, but it also contains a number of webinars and tools for farmers to interpret this. So it's been widely accepted by farmers and the seed industry, and for each of the regions in Victoria and Tasmania, and we're hoping to see an expansion into dairy regions of New South Wales in the next update. Farmers are given an overall ranking of performance relative to the base case. And so this base genetics here is an old variety Victorian, which hangs into the industry. First released, as the name suggests, here in Victoria in the 30s, so 1930s. And you can see here that the relative value of some of these novel genetics, and this is actually in predicted dollars per hectare per annum. We don't put the dollar sign because we're not asking people to do cash flow budgets based on it, but as a relative thing, you can see that there is real value in novel past genetics for farmers to extract and utilize. So this just shows us how we take the pasture trial data that exists, we take that as a performance value, we multiply that by our economic value adjusted for the seasons and come up with an index. So nice simple cartoons, but some very good combination of economics and agronomy that underpins that. And the final slide in this case study, I guess, comes back to this point that innovation has to exist in an ecosystem of partners if it's to actually be adopted and be communicated, extended, et cetera, et cetera. So what we have with the Forage Value Index under Dairy Australia's championship is the research program is done through a project called the Forage Value Index Futures project within a program called Dairy Feedbase, which is another large research program supported by Dairy Australia and Agriculture Victoria that I'm part of, and I lead this project. We work with a data company, Datagene, who actually do the routine analysis and updates. We get data from an independent company, which is the Pasture Trials Network, and it's all brought together for the benefit of farmers through Dairy Australia. But we do the innovation, we've built the building blocks, and then we work with those partners to ensure that they are routinely updated every year and new data and new traits go into that index. The next little case study I want to snap on is how some of our work in genomics has progressed. And I work very closely with the genomics team in Agriculture Victoria at AgriBio at La Trobe University, and a lot of my work really focuses on the green end of that, so the plant end of that, whereas the team at Bundura focus on the white end or the laboratory end. And to some extent, genomic selection has really been the quite revolution in what we do in modern breeding. So unlike GMOs and genetic modification, which got a lot of publicity, good and bad, this is just sort of happening, and the scale of the impact is amazing and often not promoted. And so, as I mentioned, the initial adopters were really in dairy and beef and prime lamb, and also forest trees were early adopters of the technology in plants. And so ourselves and a couple of other groups internationally have really pioneered this technology for forage plants, and particularly in our case, perennial ryegrass at the moment. And as I mentioned previously, we're really using the whole genome sequence and the variation in it to correlate with performance. So it's very useful for traits where the variation is controlled by a large number of loci or a large number of genes. And yield is a prime example of that. There's no one yield gene, there's thousands of yield genes. So estimating the tiny effect of each of those genes to the overall outcome allows us to find those elite combinations. It's also of benefit to traits that are hard to measure and expensive to measure, because once you've developed your algorithms, you don't have to remeasure them every year. You can just reset it every few years. And it's also extremely valuable for traits that are expressed late in the life cycle. So if you're thinking about the persistence or survival of pastures, you can predict what that would be before waiting for 5 to 10 years of pasture life. And I guess there's a general consensus in the breeding world now that genomic selection is the simplest and most robust method to use genomics in breeding in all cases across all species. So it will become the baseline of any breeding program. So this just shows that what we do is we take a reference population of plants and we develop a genomic prediction equation, which is looking at the variance at each locus and the weighting of that variance. We screen some candidates and we select the parents on these breeding values, and we can turn this cycle over multiple times once we've done the basic science. I didn't want to spend a lot of time on the science here, just for the sake of time, but I just want to point out that we took the approach of doing this in a way that was working with a seed company within Dairy Bio. So that seed company happened to be Barrenbrug. So we're publishing all the basic science so that can be used by any company or companies. But what we've seen is that in the duration of 1 6-year funding cycle, we've already been able to take a 12-year cycle. So this was cartoon that I showed earlier is an example from their breeding program, and reduce that by 3 years. So we've already taken a quarter of the time or out of their generation interval. And we've done that in a way by working within their already elite populations and working with their corporate breeders and working on performance data from Australia and then New Zealand, where most of their breeding is done. So again, I mentioned this value of being able to ensure that advances in New Zealand are realised in Australia and vice versa. So we're now working to improve that further and bring in new traits, and particularly traits around nitrogen use efficiency and forage quality into the breeding program. So the final portion of my seminar today will talk about the work that I'm most actively involved now, which is phenomics and digital phenotyping. And really for me, digital phenotyping, having worked in measuring plants in the field for my career, is it's been the big bottleneck. We've been the slowest part. We've been often the most expensive part once you get over the capital cost of the new sequencing machine, of course. But what phenomics also allows us to do is know not only what happened. So if I push a lawnmower into a plot, I'll know what happened. I know how much yield there was there, but I don't know a lot about why it happened. I can try and model that, or I don't know a lot about when it happened, because I'm not there very often. So we've been working at using a range of sensors to understand plant growth, particularly this, and on a range of vehicles, both UAVs, but also ground-based systems, and to try and understand that. We've also taken the approach of using the LiDAR and sonar technology that we're most often using for yield here, and these are examples of pasture plots, to try and think about the vehicles that seed companies or agronomists might want to use. So most of our initial work was done on a sort of farm side by side with the technology bolted on the front here. We've also developed UGV or robotic systems that we think will be very useful for our work going forward with lots and lots of plots to measure. But we've also looked at putting the same sensors on the lawnmower or the mower technology that seed companies use, so that they can get the data in one pass. So they're collecting their sensor data at the same time as they're mowing their plot, and demonstrating that you get the same result no matter which platform you mount that on. So the next couple of slides are just illustrating, once we've done that work and we're confident and we're implementing that work around forage yield, one of the next traits we looked at persistence, and the survival and persistence of pastures is clearly very important, and it's a real pain to measure. So we're looking at either getting and clipping plants and estimating the amount of ground versus the amount of plant, or putting rulers down and trying to work out whether there's a plant in this gap or no plant in the gap. So the work of particularly one of current cohort of students, Chintaka Jayasinghe, has really been looking at using image-based technologies to be able to do this, and to do this more rapidly and more reliably. So what you can see here from CJ's work is a range of pre-grazing, post-grazing images, and the artificial image that comes from the computer in quantifying where is the grass and where is the not grass, and what is bare ground in this case versus what is senescent or dead grass or residue versus the green material. So again, he's been doing a lot of work to try and understand this, and to the point that this technology is now being deployed to understand and look at trials as they go through, and to see where the genetics are failing, but also understanding issues that you can see here, that there's clearly a corner of this trial where the persistence, so the amount of green, is much lower than this corner. And so when we look at that as an index, you can see if blue is good and red is bad, that there is an environmental effect happening here, that if we can understand that and quantify that, we can make more accurate selections. We've also been looking at taking spectral techniques that are routinely used in the lab now to understand feed quality and forage quality into the field. So again, this is some work of a student who I co-supervised, who was actually at La Trobe University, Shaya Smith. And Shaya took this little unit here, which is about $150,000, and really designed this light shield bucket, which enabled us to get very accurate data in 30 seconds in the paddock on plants as they were growing. So no cutting, weighing, taking to a lab, sending off. And again, you don't need to be a statistician to see that these correlations are good. The lines cluster on a straight line for protein, digestibility, fiber, and the amount of moisture in plants. So we're really looking now at being able to replace slow and cumbersome methods with these new technologies. I just have a couple more slides to go through that really are now saying, well, where do we go next? Where do we take this technology, and what will be the big breakthrough? And for us, it's thinking about breeding for future climates. And we understand, and forages are no different than other crops, that the predictions from climate change can be quite dire. We've seen to look at basically lower yield, changing in seasonal growth, reducing of digestibility, so the quality of that, and dealing with more variability. And unfortunately, we've not got a plug and play alternative. So we can't say, oh, well, if ryegrass is out of the system, we'll put something else in, because we have species we know are more tolerant of heat, but their quality is also often worse than ryegrass. So we undertook a large climate modeling study that I won't go into the detail. I just want to say that it was extremely large, looking at all of the dairy regions in Victoria, part of Tasmania and part of New South Wales, and modelling those not on a farm scale, but on a regional scale, to look at the change in forage growth and quality at 20-year intervals from the current through 2030, 50, 70, and 2090. And I guess some of the concerns that come out of this kind of work, and Brendan Cullen and Rich Eckhart have also done a lot of work in looking at this area, is that some of the analogues that we look at the future climates are actually pretty concerning. So I'm currently sitting in Southwest Victoria at Hamilton, and using a dairy city of Warrnambool, just 100 kilometres down the road from me. We're saying in about 8 years' time, on average, Warrnambool starts to look like Hamilton. So the Warrnambool folks wouldn't like that. But by the time you're getting out to 2090, it looks like Wangaratta in northern Victoria, which is not a dairy region, is a very, very different growth pattern, and it's impossible really to grow perennial ryegrass there. We've also then thought about breaking that into different seasons to try and understand that, so that once you can get the analogues for these climates, you can start to develop the genomic selection. We also looked at a range of species alternative to perennial ryegrass, and some temperate grasses such as tall fescue, Phalaris coxfoot, and also tropical grasses, kikuyu and Rhodes grass, and legumes and herbs such as lucerne and plantain. This is a big data set that we haven't published yet and hope to do so soon. But what I just want to show here is that if we look at late spring growth, and just using Gippsland as an example, you can see that and here in these graphs, blue is good, red is bad. And what you tend to see is that there is regional variability, and this is looking at perennial ryegrass, but we're seeing this sort of, by the time you get out past 2030, you're starting to see that spring, the finish of spring becomes earlier and becomes more regularly earlier. But you can see that species that exist today, such as Phalaris, and are not widely used in dairy because their quality isn't up to scratch, actually have a good yield potential in those environments. And this is looking at some of those alternatives. So again, you can see that some of the C4 grasses, that tropical and subtropical grasses, have high yield potential in that season if we could fix their forage quality. So just some concluding remarks now is that we can do things that were not possible when I started my career, which is more than 20 years ago now, but the science really started 20 years ago around the genomics of this. We've got the opportunity to think about what we've learned over 20 years in perennial ryegrass. Can we do that in the concept of new species, and can we make it a 5-year program so that we can really get those technologies into these new species better? I think it's critical to understand the market, and I can't stress this enough. So much good plant genetics and plant breeding science ends up in a seed store somewhere and not out on farms because that market understanding just wasn't there. I guess the other thing that I always say to those that are designing a breeding program is saying, is there an option already out there? So should I be trying to get a better perennial ryegrass to do a job that coxfoot would already do? And because if that's the case, there's a better way to spend your money. And I've already stressed throughout the presentation the role of partnerships, both in science, but also with industry and that industry engagement. So just a couple of things when I reflect on the sort of body and work of where we're at, is that it's critical for us to get out as scientists and talk to farmers and talk to the industry we work with. It's been a bit tough in the last couple of years, obviously, but there are webinars and other options. But there's nothing like getting out with a group of farmers in their paddocks and discussing what their needs are, and also getting them to understand that these things that are coming have a role, but how can they think about getting the tweaks they need to manage it? The other, I guess, highlight as I wrap up for me that this coordinated body of investment and work has done has been able to have us train a cohort of graduate researchers or postgraduate students in all areas of this discipline that now are out there leading science programs internationally and ensuring this technology makes its way into other companies, other breeding programs, other jurisdictions, other species. So as John mentioned, I've been involved with a lot of graduate students. I've just put a cohort of the recent ones from our faculty whose work I have touched on in the presentation, some of whom are current and some of whom have finished. Whoops. And finally, I'd just like to again acknowledge that this work and the outcomes we've delivered have only been possible due to a coordinated and consistent period of funding. So we've been very fortunate in that respect, and having these investors and partners who've been with us on the journey for all that time. So with that, I'll finish and open to any questions that people may have.
Thank you, Kevin. That was a great overview. You might want to stop screen sharing while we do the questions, and I'll invite all our participants to put questions in the Q&A, and I can invite you to come in and open up your microphone and ask the questions yourselves, or I can ask them on your behalf. So if you can put questions in the Q&A, please. I'll start while people are thinking of questions. And Kevin, I was taken with the forage value index that you were talking about and thinking, well, yeah, that must be great for the farmers. And at the risk of sort of opening a whole other seminar, it occurred to me that there might be various ways of thinking about this index. And so is it based on just yield, which is the plant that gives the best yield, or is it based on the plant that gives the most nutrition, or is it the plant that persists best in the pasture, or is it the plant perhaps that needs the least water, or is most resistant to drought, or generates the most energy? I mean, how do farmers choose, and what does that index tell them?
Yeah, so at the moment, it is the plant that produces the most yield in the right season, so the best balance. We're doing work right now and have developed the models to incorporate energy into the index, and we've developed the model to incorporate persistence into the index. The reason they're not there yet is the industry lacks the data. So the pasture trial network is just now collecting the data for variation in energy concentration of varieties to allow that to come in. So what you're outlining is really the vision of where this tool could go and should go. And a bit like the early versions of these tools in animal genetics, it starts simple and it becomes more complex. And if we context that with what's happening with dairy bull semen at the moment, there are varying indices that farmers can choose depending what their targets are. Are they chasing productivity? Are they looking for a more balanced and equivalent kind of production? The other thing I should say is that it also allows us to put traits that are not necessarily defined in the index, so that if a farmer has a particular interest in or a problem with pests, we've got a list of the endophytes, which is a fungus that grows in the plant that has some insect deterrence capacity. So,
okay, thanks, Kevin. All right, let's come to a few of the questions. I'll start with Peter Mantle. So, Peter, if you can unmute your microphone, you can ask the question. Peter, are you there? Okay, maybe I'll ask the question, and then I'll come to Mohammed Palkarandish and Rich Eckhart in a moment. So you can think about how to unmute your microphones. Hopefully you'll be able to. So Peter Mansell says, thanks, Kevin. Given a lot of the pasture is consumed by dairy cows in Australia, given a lot of that pasture has been conserved, could you make some comment on the assessment of the suitability of pasture grasses for hay and silage making?
Yeah, and so I think that's partly addressed by the question that John raised earlier, in that we think obviously yield is important and getting yield of silage, but there is variability in the quality of herbage within varieties and within species. So we're looking at being able to quantify that genetic variation and allow producers to make choice. I guess the bigger issue really is the differences between species. And I sort of alluded to this, that some of the species which are more robust in terms of future climates actually have large periods where their forage quality isn't sufficient to meet the demands, particularly of dairy cattle, and whether that's as conserved fodder or directly grazed. So I believe it will become a much more important trait going forward as we look to improve tropical and subtropical grasses to come into our agriculture, which I'm very pleased to hear, because my PhD was in the genetics of carbohydrate accumulation in grasses all those years ago.
Okay, thanks very much for that. Mohammed Palkarandish, would you like to ask your question? Can you unmute your microphone?
Thanks, John. Can you hear
Yeah,
me?
we can hear you. Good.
Oh, great. Thank you very much, Kevin. I personally enjoyed very much the seminar. I really appreciate it, therefore. I have a question about the seed shattering. Do we have some of the forage crops that you work on have a seed shattering problem? And if there are some seed shattering, does it cause any issue for seed increase and seed breeding?
Yeah.
How you're dealing with this problem?
Yeah, thanks, Mohammed, and obviously a topic of interest to you. The species, one of the reasons we have worked on the species we do, such as perennial ryegrass, is that they are very well domesticated and don't have issues with seed shattering, but many of the next potential cohort of grasses do. So one of the major grasses grown in Australia, Phalaris aquatica, actually has a couple of seed retention loci that need to be managed closely in that, and there are impediments to bringing new traits in because the wild types are notoriously shattering. But it's also a big problem, obviously, if we think about Australian native grasses, which tend and have evolved to drop seed consistently, and that makes good ecological sense, but it's not great in an agricultural environment. So, but ryegrass itself is a very domesticated grass, and it was selected in the initial, well, even before the scientific phase of breeding for seed retention.
Very much good. It would be a nice place to collaborate and work in this area together.
Okay, thanks, Mohammed. Thanks, Kevin, for the answer. And let's go across to Rich Eckhart. Rich.
Hi, Kevin. How are you going?
Well, thanks, Rich.
Yeah, my question predictably about enteric methane. There's a fair bit of demand around for forages that contain secondary compounds, phenolic compounds, increased ether extract. What's the potential for breeding? I mean, perennial ryegrass is, if you listen to some of the Southern Europeans, they argue it's quite a bland grass. And meat produced off perennial ryegrass out of Australia, they call to be quite bland because of the lack of secondary compounds. What's the potential to increase those in our current species?
Yeah, so I guess, yeah, I mean, probably I think the option there, I'd say the more realistic one is to grow the other species in a mixture with ryegrass than ryegrass per se. Just getting all the molecular engineering right in ryegrass would be difficult, I would think. It does have a range of secondary compounds due to the endophytes, but it is a reasonably bland grass and doesn't have the problems that some of those secondary compounds can have when they're at high levels. I think the approach of putting some of those species in mixture also balances off some of the trade-offs of getting the optimum concentration of some of those compounds, because they often have threshold levels at which they move from being useful to having anti-productive factors. So, and we can think of the, condensed tannin as a great example of that.
Great, thank you.
Okay, and Bill Malcolm. Bill.
Yeah, Kevin, thanks. Great talk. You make things very clear. Simple question. Why isn't Vic perennial rye yet redundant?
Because I have failed, Bill. I never give it.
I wasn't suggesting that.
No. Look, I think there's a couple of reasons why. So serious answer is the market information around the pros and cons of Vic has been pretty confused. And as you're aware, it's only recent years that farmers are actually getting objective information that shows how far behind Victorian is when it comes to yield. There is the perception that it is more persistent, and it's certainly cheaper to buy seed. So I think that enables it to hang in the market in the absence of that objective information. when we combine the forage quality information and show that it's terrible in late season when it's flowering and full of rust, it will go even further back. And so what I'd say is that once the niche that Vic fills is taken over by species like cocksfoot, and which tend to be more persistent, and there's much more nutritious cultivars of cocksfoot being bred in Europe now, I think that will be the death knell for Vic.
Yeah, thanks, mate.
So Kevin, maybe I could have the last question because we're nearly out of time. And the issue that occurs to me is what about biodiversity? So at the end of the day, does this come down to advocating that we sow huge amounts of pasture with ryegrass, or are we talking ryegrass plus lots of other species? And if it's just ryegrass, isn't that an issue for biodiversity, at least for the insect population? And isn't it a threat for pests and diseases? For example, if you have pasture, if you have paddocks full of wheat, then you risk having Yersinia, or what about diseases? What about biodiversity?
So we're certainly seeing that, and over the last probably 40 years, we've seen the real dominance of ryegrass in the dairy market, and ryegrass grown in monoculture, so not even with a legume to fix nitrogen. I think we're seeing that concept being revisited now, and very actively about saying, well, given costs of urea as well as the sustainability impacts, can we get legumes back in the system? I guess what my feeling is that we're going to see a much more of a patchwork or mosaic of pastures. And if we think about the variability that exists on any one dairy farm, so if there's 100 hectares of dairy and there's 101 hectare paddocks, there'll be 10 of those that a farmer could much more, right now today, much more suitably sow a species other than perennial ryegrass. Whether they'll sow that in the first instance in a complicated mixture with herbs and more of a species-rich ley, or whether they just have a patchwork of monocultures, I'm not so sure. It's certainly easier to manage if you've got a patchwork of species around the farm. I guess the other thing that I think we're getting a better understanding of is the role of biodiversity in the less productive areas of the farm. And so having the species that you can put the foot to the pedal and really make money from in the parts of a farm, be that dairy or be that prime lamb, that have the possibility to be very productive, and then seeing those less productive paddocks become the first paddocks with an ecosystem service kind of role. Now, that's not ideal, but I think we're starting to see that evolution of thinking.
Okay, Kevin, thank you very much. I think we're spot on 1 o'clock, so I'm going to end this now. And I'd like to thank Kevin for a really interesting, clear overview of the particular area. And I'd like to thank everyone for attending this seminar today. And I hope you'll join us on the 8th of March for the next Dean's Research Seminar, when I'm going to present my own research on mosquito-borne viral encephalitis. So from forage crops to mosquitoes and brain disease. Okay, until then, I look forward to seeing you all around the faculty or on Zoom or elsewhere. Thanks very much, and I hope you enjoy the rest of the day. Kevin,
thanks everyone.